Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Has you ever experienced that dull, persistent ache in your knee? The kind that makes every step feel like a negotiation, every morning a bit of a battle against stiffness.
Yeah, unfortunately, that's very familiar for a lot of people.
For millions, it's not just like a passing thing. It's the daily reality of osteoarthritis, a condition that can just relentlessly erode your mobility, your quality of life.
Absolutely.
But what if, and this is the big question, what if your own body, specifically that aching joint, could become its own sort of advanced pharmacy?
Oh, interesting.
Imagine a future where your knee isn't just reacting to pain, but it's actively producing its own long-term relief, offering maybe true freedom from that chronic discomfort.
That's a powerful idea.
So today we're diving deep into a really groundbreaking new approach to tackling osteoarthritis, and it uses gene therapy.
Right.
We've been poring over some recent articles and, importantly, the scientific paper detailing a first-in-human phase 1 clinical trial. And honestly, it has the medical community buzzing about its transformative potential.
It really does. The early signs are quite something.
Yeah, so our mission with this deep dive isn't just to report the facts. It's more to explore this really radical idea that we could be on the cusp of rewriting how we approach these debilitating chronic conditions. We want to understand exactly how this therapy works, what these initial findings really reveal, what its current limitations are, and maybe most importantly, what it could mean for the future of chronic knee pain for you and, well, millions of others.
And it's definitely a critical area of research. Osteoarthritis, often just called OA, it's a massive global health challenge.
Huge.
In the United States alone, over 32.5 million adults are grappling with this form of arthritis.
Wow.
And at its core, osteoarthritis involves the progressive breakdown of cartilage, that smooth protective tissue cushioning the ends of your bones.
Right, it's a shock absorber.
Exactly. And sometimes it even affects the underlying bone itself. This degeneration leads to inflammation, pain, stiffness, and ultimately a significant loss of function. It's actually a leading cause of disability worldwide.
And it's so notoriously difficult to treat effectively, isn't it? It feels like we've just been stuck in this cycle of, well, temporary fixes for ages.
That's been the frustration.
I mean, we've had things like steroid injections, right?
Yeah.
Directly into the joint. But for so many people, that relief just feels really fleeting.
You've really hit on the central problem there. Current medications injected right into the joint, what we call intra-articular medications, like corticosteroids, they get cleared from that joint cavity remarkably quickly.
Yeah.
So they offer only short-term benefits, maybe just for a few weeks, often.
Right, not a long-term solution.
Not at all. Dr. Christopher Evans, who's a lead researcher on this from Mayo Clinic, he describes this challenge really well. He notes that these traditional medications injected into the affected joint, they kind of seep right back out in a few hours. Hours. He highlights that this rapid clearance creates a huge pharmacologic barrier to achieving any kind of effective, sustained treatment for such a chronic disease.
That's like trying to fill a leaky bucket, I guess.
That's a perfect analogy. It just doesn't stick around long enough to really make a difference long term.
So, okay, if the traditional meds can't stick around, they seep out too fast. That's where this gene therapy idea comes in as a potential game changer.
Precisely.
It sounds like instead of constantly re-injecting medicine, you're basically teaching the knee to generate its own defense system.
That's exactly the core promise. It's the revolutionary concept here. The idea is to genetically modify cells right there within the joint.
Okay.
So they start producing their own specific anti-inflammatory molecules. This approach could potentially make knees more resistant to those destructive processes of arthritis for a really long time, essentially transforming the knee itself into a sort of continuous, localized drug factory.
A drug factory in your knee. That's a fascinating concept. So what exactly is the specific molecular target then? What are they trying to neutralize with this gene therapy? What's the bad guy they're trying to shut down?
Right. So Dr. Evans's lab at Mayo Clinic identified a key instigator in that whole inflammatory cascade. It's a molecule called interleukin-1. You can spell it IL-1.
Interleukin-1, okay.
Think of interleukin-1 as the joint's rogue general, maybe, constantly sending out these inflammatory signals, fueling the pain, and aggressively breaking down cartilage in osteoarthritis. It plays a really central role in driving the disease progression.
So the brilliant part isn't just finding the enemy, the interleukin-1, but then figuring out its natural counteragent, or where to block it.
You're exactly right. And fortunately, nature often provides its own solutions, its own checks and balances. The natural inhibitor of interleukin-1 is a molecule aptly named the interleukin-1 receptor antagonist. That's IL-1RA.
Interleukin-1 receptor antagonist.
Got it. And this specific molecule, the antagonist, is the foundation of this gene therapy. It acts like a natural brake. It binds to the same receptors as interleukin-1.
Okay.
But crucially, it doesn't trigger the inflammatory response. It effectively blocks the destructive actions of interleukin-1, allowing the joint environment to sort of calm down.
So if this interleukin-1 receptor antagonist is the key, the therapeutic molecule, how do you actually get its gene into the specific joint cells so they can start producing it? It's not like you can just, sprinkle the gene in there. What's the delivery system?
Right. You need a sophisticated delivery system, and that's where a very clever biological delivery system comes into play. The gene for the interleukin-1 receptor antagonist is packaged into a harmless, specially modified virus.
A virus. Okay, that sounds a bit scary initially.
It does, but this isn't a virus that makes you sick. Think of it less like a germ and more like a microscopic, custom-built delivery truck.
Ah, okay. A delivery truck.
Exactly. For this specific therapy, they use something called a self-complementary recombinant adeno-associated virus serotype 2.5.
Wow, that is a mouthful.
It is. Let's just call it a specialized adeno-associated virus, or AAV vector. It's much easier.
Okay, AD vector, good.
This finely tuned AAV delivery system, it targets the joint's synovial lining, that's the tissue that lines the inside of the joint capsule.
Right.
And it seems to target some neighboring cartilage cells too. Once inside these cells, the delivered gene basically instructs them to start producing the therapeutic protein, the interleukin-1 receptor antagonist locally, right there in the joint, for a potentially very long time.
So it bypasses that whole seeping out problem.
Precisely. This approach could potentially eliminate the need for frequent redosing, and it should significantly reduce any off-target effects you might worry about if the medication was circulating throughout your whole body.
That makes sense. It's a precision strike, delivering the instructions right where needed for potentially a long time. But the real test, obviously, is what happens when you actually introduce this into a human.
Absolutely.
Which leads us right to this crucial first-in-human phase 1 clinical trial. What was the absolute top priority for the researchers going into this initial study?
Well, for any phase 1 trial, the primary purpose is fundamentally about assessing safety and tolerability. That's job number 1.
Okay. Safety first.
Always. Researchers needed to understand how safe and how well tolerated this gene therapy, the interleukin-1 receptor antagonist delivered by that specialized AAV vector, was when injected directly into the knee joints of patients who actually had diagnosed radiographic osteoarthritis.
Then did they look for anything else beyond just safety?
Oh, yes. While safety was paramount, they also closely monitored for any, let's say, preliminary signs that the therapy might actually be working. Early hints of efficacy.
Okay, so walk us through how they actually conducted this study. Who were the participants? What exactly did the intervention involve?
Sure. So in this phase 1 trial, they enrolled 9 patients. These individuals had what's called mid-stage knee osteoarthritis.
Mid-stage, what does that mean exactly?
It means their cartilage is visibly degenerating on x-rays, according to a standard scale called Kellgren-Lawrence. There were scores 2 to 3. So they're experiencing real pain and stiffness, but they're not quite at the stage yet where they'd need a total joint replacement.
Gotcha. So real patients with moderate OA.
Exactly. 6 of these participants were female, 3 were male, and their ages ranged quite a bit, from 39 up to 66. Each patient received just a single injection of the gene therapy into one knee joint.
Just one shot.
Just one shot. And they were carefully divided into 3 groups, or cohorts, to receive different doses. They wanted to test escalating doses for safety. So there was a low-dose group, a mid-dose group, and then a significantly higher dose group of the AAV vector.
And how long did they follow these patients?
They were rigorously followed for 52 weeks. So 1 full year to monitor everything's safety, any effects, how they were feeling.
A full year follow-up for phase 1? That seems pretty decent, even if it's not, the 5 or 10 years we'd eventually want to see.
It's a good start for phase 1, definitely.
So, okay, the critical question first.
Yeah.
What were the key safety findings? That's always the biggest hurdle with any new therapy, especially something like gene therapy.
Right. And on the safety front, the results were actually remarkably encouraging. The study found absolutely no serious adverse events that were considered related to the gene therapy itself.
That's huge.
It is, and critically, no participant had to drop out of the study because of any adverse events.
Any side effects at all, even minor ones.
There were some minor side effects reported by 2 of the 9 participants. They experienced temporary knee effusions. That's basically some swelling in the knee.
Okay, fluid buildup.
Exactly.
Wow.
And that was accompanied by some increased pain. However, these resolved completely with simple conservative treatments like rest and ice.
And were these definitely caused by the therapy?
Well, it's important to note that one of these patients actually had a history of similar effusions before even enrolling in the study. So the researchers deemed these events only possibly related to the therapy. They couldn't definitively say it was the cause.
Okay, so potentially related, but mild and resolved. What about broader effects? Systemic issues. Did this stuff leak out of the knee and cause problems elsewhere?
That's another key safety point. And the beauty of this targeted approach really showed here. The therapy did not cause any significant changes in vital signs or physical examination findings, or in any standard clinical lab tests, things like blood chemistry, liver function tests, complete blood counts. Everything looked normal systemically.
That's reassuring.
Very. Furthermore, the viral genetic material, the AAV vector, it was detected only transiently in the peripheral blood, and at very, very low levels, less than 1% of the injected dose.
Wow, tiny amount.
Tiny, and it cleared out of the bloodstream completely within about a week. This strongly suggests the therapy largely stayed put, localized right there in the joint, which is a huge safety advantage.
That's a really good sign it stayed where it was supposed to. Okay, what about the immune system? Our bodies are pretty good at recognizing foreign things, like a virus, even a harmless delivery truck virus.
An excellent and crucial question for all gene therapies. So most patients did develop neutralizing antibodies to the adeno-associated virus vector.
Antibodies. Is that bad?
Not necessarily bad. It's actually a common and expected response. Your immune system recognizes the viral vector, even if it's harmless, and makes antibodies against it. This was seen particularly in the mid and high dose groups.
Okay.
However, and this is highly significant from a safety perspective, no cell-mediated immune responses were detected.
Cell-mediated, what does that mean?
That means the T cells, a specific type of immune cell, weren't activated against the vector or the cells producing the therapy. Aggressive T cell responses can sometimes lead to problematic inflammation or even rejection in gene therapies.
Ah, I see.
So the fact that they didn't see these T cell responses is a very positive safety indicator. It suggests the therapy didn't trigger that kind of potentially harmful cellular immune reaction, which has been a major concern in the gene therapy field historically.
Okay, so the safety picture looks, well, surprisingly clean for such a novel approach.
It really does, based on this initial data.
But then there's always the next question. Was there any preliminary evidence, any hint that this therapy actually worked? Did it reduce pain? Improve function? Did those little pharmaceutical factories in the knee actually start producing the goods?
Right. And it's vital to remember this was primarily a phase 1 safety trial. Efficacy wasn't the main goal, but they did observe some highly promising preliminary efficacy signals, signs suggesting the surgeries were indeed operational.
Okay, like what?
First, and this is a really critical piece of biological evidence, the levels of that anti-inflammatory protein, the interleukin-1 receptor antagonist, in the synovial fluid, the joint fluid, they significantly increased in all 8 patients from whom they could actually aspirate or draw out fluid.
All 8. That's consistent.
Very consistent. And what's more, these elevated levels remain sustained for at least 1 year.
The whole duration.
The whole duration. Generally, the higher doses of the therapy led to greater increases in the production of this therapeutic protein, which also makes biological sense.
That sounds like direct, compelling evidence that the gene was delivered, it was turned on, and it was making the protein as intended. Right there in the joint.
Exactly. It shows target engagement and biological activity.
And did that biological activity translate into patients actually feeling better? Did their symptoms improve?
Yes, it seems it did, at least preliminarily. Patient-reported outcomes, these were gathered using standardized, validated questionnaires like the numeric rating scale for pain.
Okay, like 0 to 10 pain.
Right. And also the Western Ontario and McMaster University's Osteoarthritis Index, that's known as WOMAC, which measures both pain and physical function.
WOMAC, got it.
These scores showed meaningful reductions in pain and definite improvements in joint function across the group. Now, there was some individual variability, as you'd expect,
Yeah.
but it's important that all patient scores improved during the study period. And crucially, no patient had higher pain or worse WMAC scores at the end of the study than they did when they started.
That's pretty positive. No one got worse, everyone got somewhat better.
Exactly. And in fact, there was one patient who started with particularly high pain scores and very low function scores.
Uh-huh.
And by the end of the study, their scores had normalized significantly. That's quite a compelling individual response, although just one person, of course.
Still, that's quite a powerful anecdote.
Yeah.
What about structurally? Did they see any changes in the joint itself on scans? Like, did the cartilage look better, less degeneration maybe?
They did look at that using both X-rays and magnetic resonance imaging, or MRI. The imaging showed little to no progression of the actual osteoarthritis disease in most patients over that 12-month period.
So it didn't get worse?
It didn't seem to get worse, or at least not rapidly. Now, it's actually quite challenging to detect subtle structural changes, good or bad, in just a single year with OA. It's a slow-moving disease, usually.
Right.
So the fact that things looked stable is generally consistent with what you might expect over that time frame anyway, but it's certainly a positive sign. It suggests that therapy wasn't causing any rapid deterioration and, potentially might have been helping stabilize the joint environment.
This all sounds incredibly promising. I mean, really offering a potential new horizon for a condition that's so debilitating for so many.
It really does feel like a significant step forward.
But as with any early stage trial, there've got to be limitations, right? What were the main caveats with this particular study?
You're absolutely right to ask about limitations. It's crucial for interpreting these results correctly. As a phase 1 trial, it inherently had a very small sample size.
Only 9 patients.
Exactly. Only 9 patients total. This limited number means you just can't draw strong, definitive conclusions about efficacy. It's just not statistically powered for that.
Right.
And it also made it challenging to discern clear differences between the low, mid, and high dose groups, partly because there's always natural patient-to-patient variability in how people respond.
So small numbers mean you can't be absolutely sure it was the therapy causing all the improvements seen. Could other factors be involved?
Precisely. And that leads to another significant limitation, the study's open-label design.
Open label, meaning?
Meaning both the patients and the researchers knew who was receiving the active gene therapy. There was no placebo group, no group getting a sham injection.
Ah, okay. No comparison group.
Right. And a placebo group is absolutely essential in later stage trials to definitively prove effectiveness and to account for the well-known placebo effect, which can be quite strong in pain studies.
Especially for pain, yeah.
Without that placebo comparison, we can't say with certainty that the observed improvements were solely due to the gene therapy itself.
Okay, that's a big one for future studies then. What about the patients themselves? Did their individual situations or other treatments affect the results?
Yes, potentially. A couple of things there. Some patients actually entered the study with relatively low baseline pain scores to begin with.
So less room for improvement.
Exactly. If someone's starting pain is already fairly mild, it's just harder to measure a large reduction. Also, patients were permitted to continue taking their pre-existing medications for their osteoarthritis throughout the trial, things like standard pain relievers.
Oh,
This could potentially complicate the interpretation of the symptomatic improvement seen because it makes it harder to isolate the specific effect of the gene therapy on top of whatever else they were already doing.
right. You can't fully tease apart the effects. And what about the follow-up time? You mentioned a year. Is that really long enough to know if this is a truly long-lasting solution or if it's actually modifying the disease itself?
Not really, no. The 52-week follow-up period is certainly a good start for phase 1 safety, but it's simply not long enough to definitively determine if that gene expression, the protein production, and the symptomatic improvements will persist beyond 1 year.
Or for 5 years
Yeah.
or 10 years?
Exactly. And perhaps more importantly, it's not enough time to assess if there are genuine long-term disease-modifying effects. Meaning, does it actually slow down, halt, or maybe even reverse the underlying cartilage breakdown over many years?
Those are the multi-million-dollar questions, I guess.
They really are, and those require much longer studies. Finally, just a technical point, the study design didn't allow the researchers to precisely identify which specific cell types within the joint were actually transduced or took up the adeno-associated virus vector. Though preclinical work, studies in labs and animals before the human trial, strongly suggested it primarily targets chondrocytes. Those are the cartilage cells, and synoviocytes, the cells lining the joint, but they couldn't confirm that directly in these patients.
Okay, so limitations acknowledged, but still a very promising start.
Mm-hmm.
Looking at the big picture, what are the future implications here? Where does this potentially groundbreaking research go next? Especially given Dr. Evans's strong statement that this could revolutionize the treatment of osteoarthritis.
Well, Dr. Evans's enthusiasm seems well-founded based on these initial results, and he truly believes this could be transformative for patients.
Yeah.
Building on these promising Phase 1 findings, the research team has actually already completed a larger Phase 1b study. This involved more patients, likely gathering more safety data, and perhaps refining dose levels.
Okay, so they're moving forward already.
They are. And to really drive this project forward, from an academic lab setting towards actual potential clinical use, Dr. Evans has co-founded a company. It's called Genesence.
Genesence.
And according to recent reports, they are currently in advanced discussions with the Food and Drug Administration, the FDA, here in the U.S.
About what?
About launching a pivotal Phase 2b, Phase 3 clinical trial. This is the absolutely crucial next step.
Phase 2b3.
Yeah.
That sounds serious.
Yeah. It is. These are the larger, much more robust trials. They're designed specifically to evaluate the therapy's effectiveness on a broader scale, comparing it directly against a placebo and following patients for a longer duration. This is the kind of data the FDA needs before potentially considering approval for wider use.
So while these early results are definitely exciting and represent maybe a monumental first step, the really rigorous work, the proof needed for broader adoption and getting this to actual patients, that's still ahead in those bigger multi-year trials.
Absolutely. There's still a journey ahead. Future, larger trials will need to definitively confirm both the long-term safety and the robust efficacy.
Right.
They will certainly need to include those placebo groups for comparison to generate really definitive data, and they'll also likely aim to identify reliable biomarkers.
Biomarkers, like what?
Those are measurable biological indicators, maybe something in the blood or joint fluid, or even specific changes on MRI scans that could truly tell us if the treatment is modifying the underlying disease process, not just helping symptoms.
I see. Objective measures of disease change.
Exactly. And patient selection might also become more refined in these future studies. Perhaps they'll focus on enrolling individuals who have more inflammatory forms of osteoarthritis.
How would they know that?
Well, that might be indicated by specific imaging findings, or perhaps by certain blood biomarkers like elevated levels of serum C-reactive protein or CRP, which is a general marker of inflammation.
Okay.
This kind of targeted approach could help ensure that the right patients, the ones most likely to benefit, are receiving this innovative therapy, potentially maximizing its impact.
This whole deep dive into gene therapy for osteoarthritis, it really does offer a compelling glimpse into a possible future. A future where chronic pain could be managed not just symptomatically, masking the pain, but maybe at a truly fundamental level.
It feels like a potential paradigm shift.
Yeah. While these findings from the Phase 1 trial are definitely early, they provide such a highly promising, novel way to attack the disease, literally transforming the very cells in your joint into these tiny pharmaceutical factories, working tirelessly, hopefully, on your behalf.
It's quite a profound shift in thinking, isn't it? Moving away from just constantly remedicating towards potentially re-engineering the body's own capabilities.
It really is. I mean, just imagine a world where a single injection could potentially offer years of relief from debilitating joint pain.
That would be life-changing for millions.
Absolutely. And this isn't just about, a new drug in a syringe. It's about fundamentally re-engineering the body's own response to disease, basically giving it better instructions maybe to help heal itself. And it makes you wonder what other chronic conditions might eventually benefit from this kind of innovative approach, this idea of sustained localized gene therapy.
That's the really exciting long-term question.
Could this paradigm shift eventually apply to things like chronic back pain, maybe? Or even certain neurological disorders where getting therapeutic molecules delivered continuously to a specific spot is currently a massive challenge.
The potential applications could be very broad if the technology proves safe and effective over the long run.
It certainly gives us something truly fascinating to keep a close eye on as this science continues to evolve.
Absolutely. Exciting times ahead in this field.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.